perform replication studies, leading many of our common medical practices to be based on a single study, which might unknowingly claim false conclusions due to various forms of bias and statistically predictable error. Second, researchers are incentivized to preferentially design their studies and data analysis in such a way as to maximize the chance of a positive result. This results in a practice called data dredging, with a catchy term, p-hacking. One form of p-hacking includes investigating tons of secondary outcomes, since the greater the number of outcomes examined, the greater the chance that statistically predictable error will result in something appearing to be statistically significant, even if it's not a true effect. Another form of p-hacking is to not define your sample size at the outset, and instead collect data just until the results happen to dip below that magical p less than 0.05 cutoff. When the data collection was continued, the p-value can usually rise up higher again. One very common practice which spans the boundary between p-hacking and legitimate science is called post-hoc analysis, in which a hypothesis is generated and tested after data has been collected. The scientifically valid approach to post-hoc analysis would be to use such an after-the-fact hypothesis as the basis of a new experiment. After all, creating new hypotheses based on new observations is an essential step of the scientific method. Unfortunately, in practice, many clinicians take the post-hoc analysis as a definitive answer to a clinical question, and many are never subjected to new experiments. And we have no idea how many hypotheses the researchers examined after the experiment was completed before finding one that fit the data for them to include in the paper. Regarding the need to uncover unexpected conclusions in order to be published in more prestigious journals, this incentivizes some researchers to preferentially explore novel or paradigm-shifting hypotheses, which are less likely to be true, even if the evidence appears to show otherwise. And media attention is a non-negligible factor. I could create an entire video series on the role of mass media in promoting bad science. It's incredible how bad newspapers and television networks are at reporting health news. On any given day, you can scroll through the health section of your favorite news website and find articles reporting on new studies. This obviously creates publicity for the authors and for the institutions, and creates ad revenue for the media companies from user clicks. But almost all the studies reported in mass media are garbage. They are small, p-hacked, and tested unbelievable, out-of-the-mainstream hypotheses. Any doctor with experience in evidence-based medicine can very quickly recognize these studies as the trashy clickbait that they are. Unfortunately, news companies, they don't have EBM-experienced doctors on their payrolls, and journalists' knowledge of the methods and conclusions of the studies are often based just on press releases rather than the actual journal articles. Now, the most reputable scientists are not looking to have their research discussed in Oprah magazine or on the doctors. But there's actually not a clear black and white distinction between those who are excited to see themselves quoted on News Today and those who are excited to be interviewed on the Today Show. Leading publicity of all forms influences how clinicians present data and even how they choose which clinical questions to ask. So far, these conflicts of interest have focused on the interests of academic researchers, but there are less savory conflicts of interest out there, namely the financial incentives of the pharmaceutical and medical device industries. Drug companies in particular, they literally have billions of dollars riding on the outcome of a single study. That is a lot of incentive to be sure that your study shows a favorable outcome. How do they do this when the FDA does have oversight over any clinical trial that a company plans to use in an application for drug approval? First, a trial protocol may be ghostwritten by employees of a pharmaceutical company and not the published trial's official academically affiliated authors. That's not to say that the protocol isn't being accurately reported to the FDA or within the final paper, but the company will be more knowledgeable of the tricks of how to design protocols to maximize the probability of a positive result. Second, the pharmaceutical industry does the minimum necessary that's required for FDA approval and no more. If the FDA will be satisfied by a trial that tests a drug for a very specific indication in a carefully selected population with many exclusion criteria, why would a company ever fund a trial